Thank you, everybody, for joining our webinar today. As you've just seen, the webinar will be recorded, so please keep that in mind. Thank you for joining this webinar by the Center for Analytics. We're very pleased today to welcome Dr. Clement Ma and his lab, who are located at the Center for Addiction and Mental Health, CAMH. And we'll be speaking later during the webinar about their project related to accelerating mental health precision trials. But before we begin, I want to introduce myself. I'm Francis Jeanson. I'm the head of the Center for Analytics at the Ontario Brain Institute. And I wanted to first acknowledge, this land, its nature, and its people. In particular, the Anishinaabe, the Mississaugas of the Credit, the Wendat, the Haudenosaunee, and every other nations recorded or unrecorded who have taken care of this land long before us, land which we must continue to take care of together. So I'll start with a bit of background on the Ontario Brain Institute, or also known as OBI, and the Center for Analytics. So we'll have about 10 minutes to give a bit of that background, and then we'll jump on with Dr. Clement Ma's lab on their presentation, which will be about 30 minutes. We'll then have time at the end of the webinar to take questions from the audience, and you'll be able to either unmute yourself or post your question in the chat. So let me begin with a bit of this background on OBI, what we do, and the Center for Analytics. So the Ontario Brain Institute is really aimed at accelerating discovery and innovation. We're a not-for-profit organization, and we really foster collaborative research across multiple specialists in Ontario, in Canada, and even beyond, to really come together and work on important research questions related to brain disease. And we work closely with these groups to also collaborate with innovators from industry and other labs, as well as with patient communities, so that we can really look at how do we embed these innovations stemming from research and innovation into care, so it can really have an impact on improving patients' lives that have a brain condition. Over the years, we've developed major research initiatives, and these are sort of shown here in this graph, where you have these 5 major circles showcasing major research program areas that are integrated. And as you can see, they're connected to a large number of institutions across Ontario, Canada, and beyond. CANBIND is focused on major depressive disorders. We have the PON network focused on neurodevelopmental disorders, the CPNet network focused on cerebral palsy, and more recently, the CALM network focused on multiple mental health in children and youth. And finally, the Transcendent Network focused on concussion in children and adults. So these are large programs that come together and work on major initiatives, and they collect quite a bit of data. And this data is centralized on our platform called BrainCode, which you see here sort of in the middle. So BrainCode connects 40+ institutions. We have over 1,000 users. We've really focused on standardizing the data so that we collect common data elements across these initiatives. We enable data federation, also linking of these data, so we can regroup more rich data from across the participants that are part of these research studies. And we fundamentally enable data analysis. So this really creates a wonderful data discovery opportunity. We have data across these brain disorder areas that you see here through the purple columns that span across multiple modalities. So very rich data being collected that include elements such as clinical data, which often are in the form of a standardized measure or other survey. We also have a lot of neuroimaging data. So this is where participants will have potentially their brain scanned. And we could also collect other forms of imaging like CT scans or even EEG. A lot of genomics and molecular data are also being collected on the platform. And more recently, we're seeing an emergence of wearables and other sensor data being collected, which could really be beneficial for monitoring and tracking and predicting outside of the clinic, so when patients are at home or elsewhere. As a result, this data has been centralized on BrainCode, and we have a platform that now enables the researchers to collaborate. We also enable access to third parties around the world to make use of this data so that they can better understand brain conditions and ultimately come up with perhaps new techniques to detect, predict, and even potentially treat these disorders. So this has created a real data discovery opportunity, and we have a number of these data sets coming from these programs that have now been released on BrainCode. And you'll see here a list of these studies and these clinical modalities that are available, which also spans across imaging, molecular, and other forms. There's a link at the bottom here to our platform, braincode.ca, and you're welcome to visit and learn more. However, over the years, we've really identified a bit of a gap in our capacity to work with this multimodal data and also help labs and companies that are in their early stages to accelerate their technology and the development of this technology and validation of their algorithms. And we've also seen that there's been a bit of a gap in terms of support on navigating how to translate these innovations so that you can really have an impact on patient care. So this is where we've created the Center for Analytics to really focus on these core areas. And so, the first pillar for the Center for Analytics has been really around developing neuroanalytics skills to help build skill sets that can work with multiple data modalities, but also work on complex, problems that can have an impact on patient health. We also help access more modern compute so that these projects can leverage modern types of computing, such as GPUs or high memory machines that enable really the modeling of more sophisticated tools. Ultimately, we want to help translate these innovations into care. And so we can provide additional support with validation of these tools and these techniques. We can also help navigate the complex pathway towards regulatory and privacy compliance. And ultimately, we can help even productize these tools into a production type environment that can be tested in the real world, perhaps with live clinics or even patient communities. So this is a webinar series, and I wanted to highlight a little bit some of the webinars we've had so far during the course of the year. And today we will be, we're excited to invite Dr. Clement Ma and his team to present on their work on accelerating mental health precision trials. Each of these webinars relate back to our major analytics initiatives with the Center for Analytics, which are really aimed at addressing these three major pillars: the skills development, the acceleration, and the translation. So this is a very great opportunity for these labs and startups to benefit from our support, but also for us to learn from them and build a community of practice that will come together and gain skills from what everybody is doing in this space. And as it's mentioned here at the bottom of the slide, we have a third cohort that will be announced soon, so stay tuned for that. We have resources available online that you can reach. We have methods tutorials that are generated out of these analytics initiatives, and these are posted on our GitHub repository here, and we have a QR code you can scan and visit. We also have these webinars that are recorded, posted on our YouTube channel, and this is the other QR code on the right that you can view if you'd like to see the other webinars we've recorded so far. So that's it for my bit of background on the OBI and the Center for Analytics. I will now introduce our guest speakers today. So let me just stop sharing this for now. So I'd like to welcome Dr. Clement Ma. He is the scientific head of the Biostatistics Core Service at the Center for Addiction and Mental Health, CAMH. He's also assistant professor of biostatistics at the University of Toronto. Clement and his lab are creating advanced methods and tools to better design mental health clinical trials for more reliable outcomes. I'd also like to welcome Aoqi Shi, who is a PhD student at the University of Toronto and a research trainee at the Center for Addiction and Mental Health, as well as Demin Chen, who is also a PhD student at the University of Toronto and a research trainee at the Center for Addiction and Mental Health. So thank you all for joining today. I'll, I think, pass it on now to, I think, Desmond, who is going to start us off.
Hi, Francis. Thank you for the introduction. I think I will start with the presentation first.
Yes.
Can everyone see my screen? Okay. Well, okay. Hi, everyone. Good afternoon. Thank you for joining us today. My name is Aoqi. I will be talking about the general framework and main concepts for our project titled Accelerating Mental Health Precision Trials. So before delving into the precision approach, I want to give a brief overview of how typical care is delivered, which is the one-size-fits-all conventional approach. So what do we mean by that? Conventional care is rule-based and population averaged. We start from guidelines built from large trials and follow protocolized pathways that work for most of the people with this diagnosis. So the circle here with many dots show one patient population living with the same disorder. Without further stratification, many different individuals treated with the same standardized path because it works on average for most of the people. This is reliable and scalable, but average optimal is not always individual optimal. Different people may have very different response to the same treatment. As you can see to the right side, the purple group has good response. The yellow group has inadequate response, and the gray group even have no response. This is a gap that precision approach addresses. So conventional care gives us a good foundation for most of the people, and precision helps us decide which option is the right for this single person. Precision uses individual signals, such as biology, imaging, or other person-level features to individualize the prescription that the patient is most likely benefit from. Now we moved from average to person-specific. So we still have a crowd with the same disorder. The left circle is mixed crowd, but they may have very different underlying biology. So in the middle, we measure a high-resolution signal. This could be a gene panel, an imaging phenotype, or the risk score from the routine data. This measurement will reveal which subgroups with different likelihoods of benefit and whether they have very different toxicity risks. So lastly, on the rightmost step, we act on the information we gathered from the biomarker, and then we assign different subgroups into different treatments. We don't throw away the guidelines we have during the guidelines for the conventional approach, but instead it helps us to choose among guidelines acceptable options for this person and adapt earlier in the precision approach. For example, in the breast cancer, if the tumor is HER2 positive, then we start with HER2 targeted therapy. But if it's another subtype, such as triple negative, then we choose a different approach. So they have the same disease label, but the biology splits them into different groups, and then the choices for the approach will change. So precision tells us which option should fit a person. But how do we know an option truly works and it's safe? So we need failed tests. This is where clinical trials come in. A clinical trial is a study in human participants that evaluates the safety and efficacy of one or more interventions on health outcomes. The figure to the right is an example schema of a two unparalleled randomized trial control design. We first enroll eligible participants. Then we randomly assign all the participants either into the control arm, which is usually the standard care or placebo, or to an experimental arm, which is usually the new treatment. So the randomization here helps balance known and unknown factors. So differences we observe in the efficacy outcome can be contributed to the intervention rather than who happened to enroll in the study. Then we follow the pre-specified outcomes or the response of interest. Usually is the symptom outcomes, the survival, or the adverse events. So if the experimental arm really perform better than the placebo, then it becomes the new standard. Sometimes for everyone, the whole population, or sometimes is for specific biomarker subtype groups, which are identified by the precision medicine. So why do we need to run clinical trials? There are 4 reasons. The first one is to improve the treatment. Trials tells us whether a new option actually helps more than the current standard method. For example, adding a targeted drug to the standard therapy in a biomarker-defined cancer subgroups will improve the response and become the new standard. The second one is to prevent disease. Prevention trials test vaccines, screening strategies. Think of the vaccine efficacy trials that show fewer infections and fewer severe cases compared with the standard placebo method. So the third is to improve diagnosis. Some trials evaluate whether a new test or imaging method changes clinical decisions and outcomes. And the fourth one is to improve the quality of the life. An example is a new pain control method or behavioral intervention that reduces symptom burden in the patient groups. So trials establish what works, what prevents illness, what helps us detect and classify better, and what actually makes life better for patients. So I want to talk about the motivation and objectives of our current study. Here is the problem we're trying to solve. Of all psychiatry trials have a lower than average success rate. Historically, only a small portion of the phase 1 programs make it to approval, about 7.3%. So we want to have a faster trial duration and also make the trial more efficient. So first, precision psychiatry trials designs can help by prospectively using patient signals that predict response and take into account the patient heterogeneity. Second, most psychiatry trials currently still use a standard parallel group randomized trial design, but those are fair, but they leave the power on the table. So more adaptive, more novel designs can help us to faster the trial and make it more flexible. Third, investigators need better data to estimate effect size for precision trials. Effect size is a very important parameter in the trial design. So if we do not have prior information and we make a wrong estimate on this, we may have an underpowered study. To address these problems, this is our objectives for the current study. The aim one is to build AMPT, an open source planner for precision trial designs. We can choose different designs, enter parameters, and get power and sample size. The aim two is to pilot the lookup effect size tool to explore and estimate treatment effects for different diagnostic or biomarker groups using the CAMH brain code data. So left kind of provides the inputs, and AMPT turns the inputs into a design. So we have set up why heterogeneity matters. And in this section, we want to show how biomarker design uses that heterogeneity. A biomarker is any characteristics that we can measure and tells us about the biology or response to an intervention. It can be molecular, imaging, physiological, or even digital. So in oncology for the breast cancer, HER2 status predicts benefit from HER2 targeted therapy. So with this information in mind, let's see how each design uses the biomarker in practice. So due to the time limitation, in this presentation, we will only review 2 trial designs that incorporates the biomarker. They are randomized word design and enrichment design. We will compare 2 treatments and incorporate a single binary predictive biomarker. So randomized word design means we enroll the participants, we measure the biomarkers, and we randomize everyone to either control or experimental treatment. The biomarker assessment can be done before the randomization or after the randomization, as long as it is recorded for each individual. So we then classify people into either marker negative or marker positive based on the biomarker assessment. So they will all then be randomized to either control or experimental. What is the difference is how we assess the efficacy. So we can assess the efficacy in two ways. Either we assess the efficacy for the overall group, or we assess the efficacy for the specific subgroup, for example, the marker positive subgroup, if we believe the effect may be larger in this specific group. So this design is used for where we are not so sure about whether the marker is truly predictive, because it can provide us with a clean overall estimate of the effect size, the efficacy, and it allows us to explore the specific efficacy in a specific subgroup. So here is a simple real-world example. This trial asks whether nicotine metabolism rate predicts who benefits most from bupropion. So the biomarker here was pre-treatment 3-HC to cotinine ratio. It is a measure of how fast someone metabolizes nicotine. So design-wise, participants are randomized to bupropion or placebo, and everyone had a biomarker measured. So results show that the fast metabolizers had a higher quit rates with bupropion compared with placebo. So now let's see what happens if we have a very convincing biomarker so that we can focus the trial on that specific subgroup. So in an enrichment design, we screen for the biomarkers and only randomize marker positive patients. So the marker negative patients are not enrolled in the main efficacy comparison. They're excluded from the main trial. The logic is efficiency. If benefit is expected almost entirely in the positive group, concentrating randomization in this specific group increases power and reduces exposure to the treatment, unlikely to help the group in the patients in another group. So this is a real-world example, a phase 2 trial of donanemab in early Alzheimer's disease. This study enrolled patients with early symptomatic Alzheimer's disease and used a PET scan biomarker for the tau deposition. Patients with an intermediate range of tau signal, range from 1.1 to 1.46, are considered biomarker positive and are randomized to either the placebo or the experimental arm. Those outside the range are considered biomarker negative, and they were excluded from the main trial efficacy comparison. So the question was whether targeting that biomarker-defined subgroup improves the outcome. So the trial showed better cognition and function at 76 weeks for donanemab versus placebo in the enriched population. So this is the main framework and some concepts of our study. And now I will turn to Desmond for more concrete examples and live demo.
Okay, thanks, Achi. So I will take it over. So I will take it over to discuss about the live demonstration and the app itself. And so our app is called the AMP platform, which is Accelerating Mental Health Precision Trials. We will develop an interactive web-based AMP platforms using the R Shiny interface. We integrated all the R pipeline that we had, our group have previously produced for all the trial designs, which could analytically or use simulation-based methods to do power or sample size calculation given user pre-specified trial parameters. And also, user can easily download the output summary report with a study schema and also detailed text description with a click of a button, and it's in a Word document format, which can be easily copy and pasted into trial protocols. As you can see to the screenshot on the right, this is a screenshot of the prototype. Okay, let's briefly talk about the live demonstration example we will be talking about Okay, so it's the monitoring versus controlling mild and moderate Alzheimer's disease patients. So first of all, what is a Alzheimer's disease? It's a dementia and also a form of a progressive brain disorder. It's caused by a large chunk of proteins that were clogged up in the brain that were not able to be metabolized out, which this kind of buildup will cause brain cell damage. And if you take a look on this cartoon to the left, there are two cross sections of the Alzheimer patient's brain, where in the mild or the early stage of the Alzheimer's disease, you can see that the brain tissues are not shrinking too much. But when it arrives at moderate stage or middle stage, you can see the brain starts to shrinking, which will eventually lead to memory loss, confusion, and other cognitive impairment. So Mameting, on the other side, it's a approved drug for moderate to severe Alzheimer's disease. It will regulate the harmful chemical production in brain. It will stabilize the symptom, slow cognitive decline, and also finally improve the quality of life for Alzheimer's disease patients. Okay, so given our hypothetical example here, if I were a trial design researcher, I'm deciding between whether I should use randomized all design or enrichment design for this Mameting trial. If I were to use a randomized all design, we would have to enroll the Alzheimer patients, and regardless of their Alzheimer disease severity or the stage, we randomize everyone and then assigning them either to control or Mameting treatment based on the randomization. Then if I were to use enrichment design, in this case, I would enroll all the Alzheimer's disease patients, but I will have to screen for the Alzheimer's disease stages for these patients. If they were at their early stage or only have mild Alzheimer's disease, they will be excluded. And if they were at moderate, they have moderate Alzheimer's disease, they will join the trial, and we'll randomize the subpopulation of those that are at moderate stage. And so what is the biomarker and outcome for this example? So for the biomarker, we partition the participants into two groups. Moderate Alzheimer's disease or mild Alzheimer's disease, where the moderate Alzheimer's disease patients are partitioned into biomarker positive, while the mild Alzheimer's disease patients are split into biomarker negative. And the outcome measure used here is called neuropsychiatric inventory score, which is used for comparison. So the neuropsychiatric inventory score is a measure of 12 neuropsychiatric symptoms, where each one of them will be given a score from 0 to 12. So the total NPI score is ranging from 0 to 144, which was the highest score indicating a worse condition for the patients. Here, based on some of the previous literature, we found out the standardized effect delta or standardized change for the moderate Alzheimer's disease patients that used Mameting was a decrease of 0.29 standard deviation, while for the mild Alzheimer's disease patients is 0.08 standard deviation and 0 for control. And then let's take a look at the app. So if I were to design this, so when you log into the app, you will first see the landing page. So as Archie have discussed earlier, our app can compute several trial designs, but not limited to randomized out and enrichment. But for the time limitation, we're only demonstrating randomized out and enrichment for today, which is under standard design. Okay. let's start with randomized all design. So I will select randomized all design. And then in this case, because I were a trial design researcher, I need to know the budget for my trial design. So I need to know the sample size, which is directly correlated to the budget. And here I set my targeted power to 80%. So the power here means the chance of observing an effect when it truly exists. And we set it to 80%, which is the standard. And then given by previous input, the standardized effect size for M plus patients or the was 0.29 standard deviation decrease for moderate Alzheimer's disease patients, and then 0.08 standard deviation decrease for mild Alzheimer's disease patients. And here we assume the biomarker prevalence, which is the probability of seeing someone with a moderate Alzheimer's disease within the recruitment. We set it to 60%. And then for the hypothesis testing, we want to try the one-sided because we're only interested on the larger part. If the effect size is larger than 0, and then for the significance level, we choose 0.025, which is more stringent. And then randomization ratio is the ratio between the arm 1 and arm 2 on the patient's number. We set it to 1. And finally, the recruitment rate, we can simply set that to 6. 4. Okay, so after all the input has been inputted, you can click calculate now. You can see a study schema right here on the top, as well as detailed text on the bottom. And the user can easily download this entire page by click of this button, and it's in Word document. So it's coming with a figure, which is the study schema, and also text. The user can easily modify and use that, reuse that in their trial protocol. Okay, so if I were to try the enrichment design with similar parameter in this case, we just have to click on enrichment, and the input should be similar. Let me double check. Yes. Then I click on calculate now. You can see the study schema is now changed to an enrichment design, as well as the text description below. Okay, let's look back to the presentation. Okay, so we have done a live demonstration, and we can compare the results from our hypothetical example. So as you can see from the enrichment design, the average treatment effect was 0.29, which is the same as only moderate Alzheimer's disease participants. But in randomized all design, it's 0.206 standard deviation. In this case, because we were the population for this randomized all design was mixed with the mild Alzheimer's disease patients, so the effect size get diluted. And also, if we look at the total sample size in here, the enrichment design had a total sample size requirement for 374, while randomized all requires 740 to achieve the similar level of power, which is 80%. We have said it. And also, if we compare the trial duration, the enrichment design is it's only taking 63 months and versus 74 months from the randomized all design. So given all this, we can conclude that the enrichment design is more efficient than randomized all because we are limiting the the trial to those to the subpopulation who are more responsive to the drug. So the differences here reflect how biomarker targeting improves trial efficiency and speeding up the entire process, and also illustrate the type of insights user can quickly generate with this app. Finally, some conclusions for this. So what are the roles of the Shiny app? It first enables comparison of power, sample size, and duration across all the designs. It provides a user-friendly interface for visualizing designs, and also a convenient, with a click of a button, the user can download all the output. It facilitates transparent, data-driven decision-making in trial planning. And it enables the investigator to design precision trials and generalize this to other psychiatric disorder. And finally, AMPT will accelerate the development of precision therapies for mental health disorders, ensuring the right treatment for the right person. And this is our team. We thank for the generous funding provided by CAMH Discovery Fund, as well as OBI Center for Analytics and Neural Analytics Initiative. Thank you. Any questions?
That's great. Thank you very much, Aochi and Desmond, for this fantastic work. It was really nice to see a live demo too, which is always a bit risky when you're doing a presentation. So thanks for that. I'll invite anybody from the audience to either raise your hand or post a question in the chat if you'd like to ask any questions for the team. While folks either post or ask questions, I'd love to follow up quickly. I saw in your last slide, Desmond, around some of the benefits. I mean, clearly being able to reduce the number of participants you need to recruit is a tremendous advantage. It also, I think, based on what you're describing, really helps target the potential therapy much more precisely. But I was curious because you have a sample size here of 374 versus 740 in the randomized. That's almost half. And yet the estimated duration of the trial is not quite half. I would have assumed it would have been half of 74 months. Can you explain a little bit that? How is that being calculated?
Yeah, so that's a great question. So it's actually a bit counterintuitive here. So because like the sample size here for the randomized audit design is 740, right? But we're basically recruiting everyone with Alzheimer's disease, despite no matter which stage they're at. But if we were to recruit this 374 Alzheimer's participants with Alzheimer's disease and only limited them to moderate Alzheimer's disease, then the recruitment rate will be lower in general in comparison if we were recruiting everyone without screening.
Right.
So yeah.
So it's essentially the sort of presence of the catchment, I guess, of these participants is going to be less likely because they have to fit the biomarker condition. Understood.
Right.
Makes a lot of sense. Thank you. Is this calculation? So I guess this is done through the platform itself that you showed earlier, and it takes into account these rates and these likelihoods, I guess, of the population being present or presenting with these biomarkers.
So the user would have to provide the recruitment rate because we're not pre-assuming any of that, because that would depend on how big of a institute they are and how much patient they're getting. So this may vary. So we let the user decide.
Right. That's great. Any questions from the audience? I'm not seeing any yet, unless I'm missing it. But feel free to either unmute, raise your hand, unmute, and you can ask your question or type it in the chat. I had a follow-up question a little bit related to the, one of the early slides, I think that Ouchi presented on the current success rate of trials being fairly low, I think in that 7%-ish, between 7 and 8%. Is there an idea as to how much better we can get through more precision trials? I guess depending on the biomarkers we find, et cetera. Do you have an idea either from the literature or from your own research as to where we might be able to get?
Yeah, that's a good question. So like the challenge here is like especially for mental health trials, the patient recruitment is the first challenge. Whether we have enough patients to conduct the trial and how fast we can recruit the participants. This can also impact the success of the trial. So that's why more advanced or novel trials and precision psychiatry can try to make the trial faster because it can provide some flexibility where we can, during the mid course of the trial, we can make some changes. And this can, maybe later we find out about that, we will miss some time to make the adjustments, but during the mid course of the trial, we make the adjustments as fast as possible, then we can shorten the trial duration. And the second is the precision psychiatry trial, because patient heterogeneity is an issue. If we target different patient heterogeneity and then specifically for a subgroup, then we can also improve the trial success rate.
Do you envision a scenario where you may have a population with multiple biomarkers and the enrichment trial becomes a little bit more complex, and so you'd have an arm with a certain biomarker, an arm with another biomarker, or perhaps a mix of two as a control? Is that something that's been explored already?
I think there may be very different designs other than the randomized or the enrichments that can then can take care of the more complex scenarios. Yeah.
Okay. Yeah, that's very interesting. I don't see any other questions from the audience yet. I did have a follow-up on, I think Ouchi, you mentioned at the beginning kind of your work on this tool with Desmond. You also mentioned the work on leveraging some of the Combine data and working towards the lookup tool for effect size. Can you comment on that a little bit more and sort of the future next steps on that?
Yes. So effect size is a very important part for the trial design, because using the planned effect size, we can decide based on our budget how many participants we will need for our trial. And so in this case, if we can, and usually there will be some numbers you get from previous trials or the pop literature that you will use to kind of design your study. But for the AIM2, we are trying to kind of for different subtypes of the patients in the combined brain code data, we can estimate the effect size for the specific group. And then you can use the effect size like as input for our AIM1 platform. Then you can plug in the effect size, then we can get more output for your trial design. So that's our next step.
That's fantastic.
Maybe I can also add, I think our project really synergizes with the other projects that's really currently hosted by the Center of Analytics. Some of the other projects are trying to identify subgroups of participants who respond better to certain treatments in all different diseases. And I know our example today was for Alzheimer's, but I think we can leverage some of the other tools that the other teams are building. They will discover the appropriate subsets, identify the biomarkers that are predictive, and then investigators can use our app to build the trials that incorporate those biomarkers. So I think we're trying to be synergistic here with some of the other projects as well, which is very exciting.
Yeah, thank you, Clement. Yeah, great point. That's very true. Yes, we had some, I think, great webinars from other members of the Center for Analytics, including Fernak Farzan's lab and Joe Garacci and Netromark, doing just like what you said. Using the data to find these markers and help kind of understand what the what essentially the I guess the population looks like, and then that can help inform sort of what you input into the tool, like the AMPT tool here, to build better trials, which is really, creating a lot of synergy, I think, between the projects, which is exciting to see. That's fantastic. We have a great comment actually from Melissa. She wanted to thank everybody for pursuing this important research. The precision medicine is really an important area, an exceptional idea. The specificity, scaling the research, and implementing it is a massive hurdle to overcome. So she was really grateful for the presentation and the work being done here. So that's great feedback. Thank you, Melissa. So I don't see any other questions. Andrew?
Yep. Sorry. So I'm Andrew from Mustard Seed, and I mostly work with startups in the space, whether it be CNS or just life sciences in general. Thank you for the talk. It was great. And my question is, do you think the primary value proposition of physician psychiatry to be in, accelerating the development of companion diagnostics to, better utilize the existing drugs, or do you think it's sort of like a deeper, mandate to use the biomarkers for target validation and development of new classes of drugs? Does the question make sense?
Yeah, that's actually a great question. I think that the main point I think you're asking about is whether we should be mainly focusing on this drug repositioning versus developing new drug. We understand that a new drug would take I think on average take like $2.6 billion to develop, and yeah, there are also some other biomarker-based identification methods that are used for drug repositioning as well. But yeah, I think those were mainly based on the omics, and some of that, I think some of the rather hot topic are regarding the using some sort of causal inference framework to find that how different kinds of symptoms there or different kinds of traits they're co-localizing with each other, and that will inform the drug developer to reposition the existing drug for this kind of trait to another one. Yeah, I hope that answers your question.
Yep, it did. Thanks.
Thank you, Andrew. Do we have any other questions from the audience? Just have a few minutes left. Looking pretty good. That's great. Well, I think if we can probably end the webinar a little bit early if there are no more questions, but I first want to thank Dr. Clement Ma, Auchi, and Desmond for joining us today and for presenting on this exciting work. I think there's a lot to learn from better trial design and how this can have a real impact on more precision health. So yeah, I think this is very exciting, and we're exciting to see also work in the aim-to very innovative thinking, I think, on the concept of leveraging existing study data and reusing that data for the purpose of improving trial design. So I think really exciting work to come. Everybody stay tuned. All the CFA projects are working also on developing methods tutorials, so we'll have some more content to share in the future, including from this project by Dr. Ma's lab. And we will be posting this webinar along with other webinars on our YouTube channel. So maybe I'll ask Auchi or Desmond to stop sharing. I'll share my screen. Thanks. Just for everybody's sake, in case I went too fast earlier, these are the QR codes, and on the right, you have the CFA webinars link here if you'd like to take a snapshot and visit these webinars in the near future. We'll be posting more of them in the coming months. So thank you everybody for joining. This is a great webinar, and we look forward to touching base again soon. I think we had the previous slide here showing our upcoming webinar with ophthalmo on November 27th. So that's just a few weeks from now. So stay tuned for that, and thank you all for joining. Have a great day.